Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic electron transport system. This is from the chapter respiration. So watch the entire video lecture to get basics out of it. So without any delay, let's start with the topic electron transport system from the chapter respiration. So let's start. So let's start with the topic electron transport system and oxidative phosphorylation. So you can see in the left hand side, that is electron transport system cycle has been given. So electron transport system, in short form, it is also called ETS. So it is the metabolic pathway present in the inner mitochondrial membrane through which electron passes from one carrier to another carrier. Okay, so ETS is the electron. Okay, so ETS means electron transport system. So it is a metabolic pathway present in the inner mitochondrial membrane through which electron passes from one carrier to another. So this is to release and utilize energy stored in NADH plus hydrogen ion and FADH2 formed during TCA cycle by oxidation. The electrons are passed on to oxygen to form water. Okay, so the electrons are passed on to oxygen to form water. Next you can see electron from NADH are oxidized by an NADH dehydrogenase. Okay, so this is complex 1. Complex 1 is NADH dehydrogenase. Electrons are then transferred to ubiquinone, that is Q, located within the inner membrane. You can see that. Then ubiquinone, okay. Then again I repeat, ubiquinone also receives reducing equivalent via FADH2, that is complex 2, that is generated during oxidation of succinate in citric acid cycle. Next you can see the reduced ubiquinone, that is ubiquinol or QH2, is then oxidized with the transfer of electron to cytochrome C, okay, via cytochrome BC1 complex, that is a complex 3. So cytochrome C is a small protein attached to the outer surface of the inner membrane. It acts as a mobile carrier of electron between complex 3 and 4. So complex 4 refers to cytochrome C oxidase complex containing cytochrome A and A3 and 2 copper centers. So you can see the names of different complex are given: complex 1, complex 2, complex 3, complex 4 and complex 5. In complex 1 there is NADH dehydrogenase. In complex 2 there is FADH2. In complex 3 it is cytochrome BC1. In complex 4 it is cytochrome C oxidase, and in complex 5 it is ATP synthase. Okay, so when the electrons pass from one carrier to another via complex 1, 2, 3, 4, they are coupled to ATP synthase. Okay, that is complex 5 for the ATP production. So number of ATP molecules produced depends on the nature of electron donor. Okay, so number of ATP molecules produced depends on the nature of electron donor. That is, oxidation of 1 NADH is equal to 3 ATP and oxidation of 1 FADH2 is equal to 2 ATP. So again I repeat, oxidation of 1 NADH is equal to 3 ATP. So 1 NADH is equal to 3 ATP and 1 FADH2 is equal to 2 ATP. Now you can see left hand side, the overall electron transport chain is given. You can see NADH plus hydrogen ion. Then it passes through complex 1, that is NADH dehydrogenase. Then it forms quinone, okay, ubiquinone. Then complex 2, that is FADH2 in succinate joins there. Succinate plus complex 2 plus Q. Okay, it gives passes through complex 3 and then cytochrome and then complex 4 and water. Okay. So in aerobic respiration, the role of oxygen is limited to the terminal stage, yet oxygen is vital since it drives the whole process by removing hydrogen from the system. So oxygen acts as the final hydrogen acceptor. In respiration, energy of oxidation and reduction is utilized for the phosphorylation. So this process is called oxidative phosphorylation. So what is oxidative phosphorylation? In respiration, energy of oxidation and reduction is utilized for the phosphorylation. So this process is called oxidative phosphorylation. Oxidative phosphorylation is different from phosphorylation. Okay, so oxidative phosphorylation is different from phosphorylation. Here light energy is utilized for the production of proton gradient for phosphorylation. Okay, so here light energy is utilized for the production of proton gradient for phosphorylation. So you can see the energy released during ETS is utilized to synthesize ATP. Okay, so the energy released during ETS is utilized to synthesize ATP by ATP synthase, that is complex 5. So ATP synthase has two major components. One is F1 headpiece, another is F0. So F1 headpiece is peripheral membrane protein complex and F0 is integral membrane protein complex. Okay, so you can see there the pictures are given. So F1 headpiece, that is peripheral membrane protein complex, site for ATP synthesis from ADP and inorganic phosphate, and F0, that is integral membrane protein complex, it forms a channel through which proton crosses the inner membrane. Now you can see the passage of proton is coupled to the catalytic site of F1 component for ATP production. Okay, so the passage of proton is coupled with the catalytic site of F1 component for ATP production. For each ATP produced, 2 hydrogen ion passes through FO from the intermembrane space to the matrix down to the electrochemical proton gradient. So you can see in the figure given below, 2 hydrogen ion is pumped, it passes through F0, clear, from the inner membrane space to the matrix down the electrochemical proton gradient. Now in this slide you can see the entire electron transport system. You can see complex 1, complex 2, complex 3, complex 4, complex 5. Okay, the matrix of mitochondrion, the inner membrane space, the outer membrane, the cytosol, the inner mitochondrial membrane, everything is given. One is inner mitochondrial membrane, another is intermitochondrial membrane. You can see the differences. And at last you can see in complex 5 ATP is produced. So student, this is all about the electron transport system. You have to memorize this, you have to draw this cycle. Thank you student. This is all about the entire way lecture. You have discussed about the topic electron transport system from the chapter respiration. So stay at home, stay safe. I am Shomujit sir, signing off for today, and again we'll meet in the next class. Thank you students.